This is the insular cortex and compulsive cocaine use. What is compulsive drug use? Well, the easiest way to describe it is the continuous or persistent use of drugs despite knowing the negative effects that it will have. What is its relation to the insular cortex? Well, the insular cortex has been linked to addiction, particularly with the urges that come with addiction. This study only focuses on the anterior insular cortex. They did a small test to see if the posterior insular cortex also had any effect in this, and the evidence was non-conclusive. The anterior insular cortex, or the AIC, is also connected to the orbitofrontal cortex, or the OFC, as well as the amygdala. In previous studies, rodents involved in self-administration drug use tend to have a resistance rate of about 30%, which the researchers in this study hypothesized that this may be a case of different stages of addiction. Why this experiment? Well, it's to see how the AIC connects to compulsive drug use, as well as possible additional ways to treat addiction. The researchers' hypothesis states that here we hypothesize that the AIC and its associated circuit may be involved in compulsive cocaine use. In the present study, using a foot shock punished cocaine self-administration procedure combined with clustering analysis, immunostaining, fiber photometry, electrophysiology, and chemogenetics, we found that the AIC glutamatergic neurons and the OFC to AIC circuit played an important role in regulation of compulsive cocaine use, which provided a previously unidentified anatomical and functional circuit mediating drug addiction. What are the methods and materials used in this? Well, the animal models used in this were adult male Sprague Dewey rats. They underwent stereotactic surgery, which is a minimally invasive surgery used to infuse animal models with viruses. The viruses that they used were injected into the AIC and the OFC. They also used electrophysiology, specifically whole cell patch clamp recordings of the AIC neurons. The specimens that they took had been incubated in artificial cerebrospinal fluid. They also used clustering analysis, which they used a machine model to measure rat compulsive cocaine use. And the resistance rats, which I'll talk about in a minute, are categorized as rats who continuously partake in compulsive drug use despite the punishment that they receive. They also use immunostaining to see CFOS staining, and the antibodies that they use are primarily mouse and rat antibodies. A continuation of the methods that they use is a cocaine administration training. The chambers that the rats were put in consisted of 2 nose poke holes. One was an inactive hole, which didn't do anything, and one was an active hole, which, when activated, would release cocaine. They underwent 12 days of 6 one-hour sessions with a 5-minute interval in between, as well as a 20-second timeout period after every active nose poke. There was a limit of 20 infusions per session to avoid overdose, and the activity of both inactive, active, futile, which is active nose pokes that were done in the 20 seconds in between the timeout session, as well as the cocaine infusions were all recorded. The punishment test was done after the administration training, It was very similar to it, with the addition of anytime one of the rats went to the active nose poke, there was an additional foot shock. This lasted for 3 consecutive days, and the activity was recorded and limited the same as the administration training. The findings from this experiment included 3 tests, which all had 2 parts each. The first was a causal link between neuronal activity and the AIC. Second was a glutamate manipulation, and the third was an OFC to AIC circuit manipulation. The experimental group received a virus that was either to induce inhibition or excitation, and the rats' resistance were recorded. In the AIC activity, the experimental group was first given an inhibition virus, and during the third punishment test, the resistance group for the experimental was recorded at 10 out of 31 resistant rats, and the control group was 10 out of 28 resistant rats. After the 6th punishment test, the control group was at 9 out of 28 resistant rats, and the experimental group no longer had any resistant rats. During the excitation test, the third punishment, the experimental group ended with 12 out of 13 resistant rats, and the control ended with 5 out of 14 resistant rats. And I should note that the reason why the researchers are both doing an inhibition virus as well as an excitation virus is because throughout all of these tests, they are hypothesizing that inhibition is what is going to slow down the compulsory drug use. That's why there is, after the 6th punishment test, there is no more resistant rats due to compulsory drug use. However, to make sure that it is actually because of the inhibition and not just correlational data, they also have to do the opposite and excite the neurons or whatever part of the brain that they are working on to just make sure that it's not correlational data. For glutamate manipulation, the process was similar as last, but instead of just in the AIC, it was specifically the glutamatergic neurons. Once again, first with inhibition. After the 3rd punishment test, the experimental group had 8 out of 29 resistant rats, and the control had 10 out of 32. After the 6th, the experimental group also had none, same as the last, and the control had similar to after the 3rd punishment test. During excitation, after the 6th test, the experimental group ended with 12 out of 14 resistant rats, and the control ended with 4 out of 12 resistant rats. The reason why they did a glutamate test specifically is because most of the neurons in the AIC are glutamatergic, and the results from this suggest that this could lead to condition diversion, and possibly even more than condition diversion, and possibly even suppress the reward feeling that you get from cocaine. Orbitofrontal cortex anterior insular cortex circuit. The OFC to dorsal striatum circuit is believed to be very closely related to compulsory behavior, and because the AIC also connects to the OFC as well as the amygdala, the researchers believed that this link could possibly have the same effects as the circuit from the OFC to the dorsal striatum, and this could possibly be the shift from controlled drug use to compulsive drug use. The OFC and AIC circuit was chemogenically inhibited in the experimental group, and that's essentially just a fancy way of saying that they put another virus into the OFC. The test, once again, was performed same as the others. The third punishment test ended with 9 out of 32 resistant rats, and the control ended with 10 out of 32. During the sixth punishment test, the experimental group once again ended with no resistant rats, and the control ended with 10 out of 32. The OFC to AIC circuit was chemogenically excited then, and the experimental group ended with 12 out of 15 resistant rats, and the control ended with 3 out of 9. Why is this data important, and why is this study important? Well, there's still a lot to learn about the insular cortex and addiction as well. And studies like this and research like this, not only does it give more insight on these aspects individually, but it also gives more insight on how they interact together, how the insular cortex could have an effect on addiction. And knowing this information can help lead to better support for addicts and treatment plans. The article that I used in the study is titled "In Orbitofrontal Cortex Anterior Insular Cortex Circuit Gates Compulsive Cocaine Use."